Here we have our report for the Gulf Specimen Marine Lab in the St. Mark's Lighthouse. On the left is me in front of the aquarium, and then on the right is me where they have their turtle nursery. Then we'll start off with some organisms with shells. On the left we have the quahog clam. And then on the right we have the leopard crab, which I thought was pretty cool. I've never seen one of them before. Then we have the left-handed whelk on the left, and then the hermit crab on the right. And I accidentally dropped this hermit crab, because it touched me with one of its claws. Then we had the fiddler crab. They had their own exhibit. There was hundreds of them in this one little area. Then we have on the right the horseshoe crab that we got to pick up and look at its underside. Then this nocturnal organism, which is known as the nurse shark, we got to feed them, and surprisingly, instead of biting at their prey, they actually suck in their food. Then we have organisms that have tentacles. On the left we have the squid that's in a preservative tank. They didn't have any live octopuses or squids. Then on the right we have the torch coral. They had these. There was a bunch of these with different colors, and they were pretty. Then we have the tropical plumed anemone. Here we have two sessile organisms. On the left we have a sea pansy, which I never knew existed, as well as on the right with the sea cucumber. Then here's some fish that we saw. On the left is a Nemo, or the clownfish, and the ocellated moray eel. There was a bunch of these, and they kept looking up at us and showing us their teeth. Then we have the long-snouted seahorse. There was a bunch of these, and they were different colors, and it was cool seeing these. Then we have the poisonous red lionfish. We got to learn about this pretty well, and about the poison and their different uses for it. And each lionfish typically has around 18 spines. Then we have the stingray. They showed us to splash our hands in the water, and they would come up and let us pet them. Then on the right we have the scorpion fish. Now we have the shark sucker that was in the same tank as this green moray eel on the right. This one kept following me around the tank as I walked around, and would show me its teeth and kind of jolt at me like it was going to bite me. Then we have the starfish. On the left we have the chocolate chip sea star, which was pretty cool to see. I've never seen one of them before. And then on the right, which I don't know the name of specifically, we have another starfish. Unfortunately, this one was broken. Then we have the scrawled cowfish. I'm not sure where it gets its name from. I'm guessing from the spots that it has on it. And then we just have the normal sand dollar. And then here we had to do 6 articles, either based on climate change or acidification. The first one is the climate change and its effect on stingrays. In this article, in their study, they found that the increase in water temperature caused embryo development to be sped up, and cause the success rate of them to have a decrease. Then the younger stingrays, they found they had a higher metabolic thermal sensitivity, and were the most susceptible to the rise in temperatures, especially in the nurseries in their intertidal habitats. And then also the higher temperatures were also found to play a role in the reduced growth rates that cause the stingrays to grow smaller in their adult stage, which made them more prone to predation and less capable to compete for their prey. The second article is on ocean acidification and its effect that it had on sea cucumbers. This article and in their study found that the post-fertilization success, that there was a decrease by 6% when the pH got below around 7.4 compared to a pH of around 8.1, which was considered normal. And then also we see in the early stages of development that with an area that has a decrease in pH with a more acidic, that it caused the developmental time to increase, which resulted in the success rate to decrease. And then also the pH area that was more acidic, the size of larvae, they saw that they tend to increase in size. And then the third article is the climate change and its effect that it has on nurse sharks. It found that their metabolic rate drastically increased with an increase of temperature, and the increase in temperature also increased how much oxygen they needed to consume. The nurse sharks in cooler temperatures had a lower oxygen consumption, which was also linked to their low metabolic rate. The fourth was on the effects climate change had on squids. It was found that the size of the hatchlings and that came from the eggs became and grew smaller in higher temperatures, which ultimately caused the hatchlings to grow smaller into adulthood. The squids were also found to be able to, the older squids were able to adapt and respond more quickly to their changing ecosystem in their climate with a rise of temperature. And then the elevated temperatures showed that it accelerated the life of squid and made them grow quicker, which increased their growth rates and ultimately shortened their lifespan. The fifth article is on the acidification and its impact on sand dollars. The percent of fertilized eggs and sperm drastically decreased when the area was more acidic. And then areas with a pH higher than 7.6, that there was a delay in development. And then any areas with a pH higher than 7.79, so around 7.8, there was a big mortality rate. And that there was variation in the sizes of larval and adult sand dollars, with the sand dollars being found in a lower pH area. The last article was the climate change and its impact on seahorses. It was found that seahorses in a warmer climate had an increased metabolic rate, but their food intake remained the same. Newborn seahorses showed the most changes and were less prone to adapt due to the increase in temperatures, and we saw that they developed metabolic depression. Then we had to do 4 articles for unique chemicals found in some of the species that we saw in the lab. The first one is the green eel. It's chemical, and it comes from the mucus that its skin secretes. The mucus contains palmitic acid and stearic acid, and this is just an adaptive trait that the eels have since they don't have fins. And so they use this when they hide in dark areas like holes and cracks and wait for their prey to swim by. The mucus covering on their skin allows them to get in small spaces to hide and wait for their prey so they can attack. The next chemical was in stingrays. The venom from a stingray spine contains a neurotransmitter serotonin and 2 enzymes. The number 5 nucleotidase and phosphodiesterase. The serotonin is responsible for the terrible pain that the sting causes, because it causes the smooth muscle to severely contract. And then the 2 enzymes are what causes tissue necrosis, which are the death of cell and tissues. The next chemical was in the lionfish. The venom contains proteins such as hyaluronidase, which causes the pain, and they also have neuromuscular toxins in them. The venom is highly concentrated, and they compare this toxin and venom to that of a cobra snake venom, and the symptoms from a sting can cause drastic change in your heart rate, abdominal pain, and can last from 8 hours all the way up to 30 days. The next, which I was shocked because I actually held this one, was a chemical in the sea cucumber known as holothurine. It's very potent and poisonous to smaller animals, but the venom can also cause blindness in humans. Now we'll move on to some of the things we found at St. Mark's Lighthouse. Some of the species that I found were the ghost crab and the blue crab. There was a bunch of the blue crabs, and the blue crab actually bit my foot. Then here's two types of birds that I found on the left. Can't really see them, but there's two black birds, which are known as boat-tailed gackles. And then on the right, I'm not sure what the name of the bird is, but it was brown with a few white spots. Then we have the sable palm tree, and then the thin-striped hermit crab. There was a bunch of these hermit crabs all throughout the beach and in the water. Here's two types of snails that I found. The one on the left was actually on a weed growing out of the sand, and then the right one, there was a bunch of these in the actual water. And then here's on the left we have a fish. There, I believe I saw these at the lab, but I can't remember the name. And then there was a tree trunk in the water a little bit, and there was a bunch of clams that we saw. Here's some of the end of the report questions. On the right, you can see a map that shows the St. Mark's River. It goes up and ends ends near northeast of Tallahassee, and it does not cross into Georgia anyway. Then the first question was, what is the salinity of the ocean? And on average, about 3.5% of the weight of seawater comes from dissolved salts, and the concentration of salt in seawater is about 35 parts per thousand. Then what was the average pH of the ocean? And the ocean is slightly basic with a pH of around 8.1. Then the average dissolved oxygen level in the ocean was just below 5. Number 4 was the 5 main cations and the 5 main anions found in seawater. The cations include sodium, magnesium, calcium, potassium, and strontium. And then the anions were chloride, sulfate, bicarbonate, bromide, and borate. Then number 5, where is the deepest point in the world's ocean? And that would be the Mariana Trench. It is located beneath the western Pacific Ocean, and it is approximately 36,200 feet deep. This was our report for the Gulf Specimen Marine Lab and for St. John's. Thank you for watching and hope you enjoyed.